feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, hello, and welcome to the National University of Singapore Yong-Loo Lin School of Medicine Healthy Longevity webinar. Thanks for joining us tonight. We have Charles Brenner. I think it'll be an exciting discussion. He'll be on in a few minutes, and please use the Q&A function to ask questions, and we'll try to get as many of those to Charles as possible. Before we start with that, though, we have Shiva, research assistant in the Center for Healthy Longevity, who'll be telling us about a recent study about cytosolic and mitochondrial TNA synthetase inhibitors that increase lifespan in a GCN4 ATF4 dependent manner.
Thank you so much for the introduction. As mentioned, my name is Shiva, and I'll be presenting a paper today by Robbins. It just so happens that Professor Brian Kennedy is also one of the authors on this paper. So previous studies that have looked at ribosomal protein deletions in yeast have shown that certain deletions have led to increased translation of the protein GCN4. And this has also increased replicative lifespan. Replicative lifespan is the number of divisions throughout the life of a yeast cell. GCN4 is a functionally conserved transcription factor that is translationally upregulated by uncharged tRNAs. It is also notable that the GCN4 pathway shown here is conserved from yeast to mammals, where it is known as ATF4. This indicates that an increase in ATF4 levels might be associated with an increase in lifespan. Since the accumulation of uncharged tRNAs is important to upregulating this pathway, researchers postulated that addition of a tRNA synthetase inhibitor would promote the pathway. tRNA synthetases aid the association of an amino acid to the appropriate tRNA, thus converting it to a charged tRNA. So, in order to maintain a level of uncharged tRNAs, it would be necessary to use a tRNA synthetase inhibitor. Going forward, researchers identified two tRNA synthetase inhibitors to test. The first drug looked at was borolidine, which is a biologically synthesized polyketide that inhibits threonal tRNA synthetase. What researchers found was that in liquid media at 80 micromolar concentration of the drug, there was an increase of doubling time without any complete stopping of growth. They also showed that in liquid media, concentrations between 40 to 320 micromolar could inhibit growth in different conditions. Overall, borolidine was found to inhibit yeast growth. Following this, researchers wanted to understand the effect of borolidine on GCN4. GCN4 is regulated at the level of translation by GCN2. To measure the translation of GCN4, they used a dual luciferase GCN4 reporter, which translates luciferase with the translation of the protein of interest. So with an increase of translation, there would also be an increase in luminescence. As can be seen in Figure C, with the deletion of GCN2, borolidine did not have any effect on GCN4 translation as compared to the wild type. This indicated that the start of GCN4 translation was completely dependent on GCN2. And as can be seen in Figures F and G, in both the wild type and GCN4 deleted strains, they found that there was a reduction in relative protein synthesis that was not dependent on GCN4. Overall, this indicates that the changes in GCN4 are solely due to translation. And measuring the RLS of yeast, researchers found that RLS was increased by borylidin at a dose that was shown to highly induce GCN4, while in the GCN4 deleted yeast, there was no increase in RLS. Mupirocin was also another inhibitor that researchers tested. With an increasing dosage of mupirocin, they found that there was an increase in the developmental delay through the larval stages, but no growth arrest. They also found that the developmental delays from mupirocin was independent of ATF4. As can be seen in Figure B, even with ATF4 deletion, similar levels of developmental delay was observed. This was repeated in solid media with the same results. What this indicated is that there was a developmental delay with mupirocin treatment that was ATF4 dependent. Both inhibitors were found to extend the lifespan of C. elegans after the developmental stage when the worms had reached adulthood. An increase of lifespan can be seen with both inhibitor treatments on the wild-type worms. However, we do notice that in the ATF4 deleted C. elegans, there is no longer any lifespan extension benefits, which indicated that the inhibitors were now ATF4 dependent. Finally, researchers wanted to understand at which point in life did mupirocin confer lifespan extension to the worms. Of course, whole life treatment with mupirocin increased lifespan the most, but the majority of the benefit came from the treatment in adulthood. This can be seen here by the difference between the lifespan extension between both figures A and B. The overall findings of the paper are as such. Cytosolic tRNA synthetase inhibitors upregulate GCN4 translation and extend yeast lifespan in a GCN4-dependent manner. They also extend the lifespan of C. elegans in an ATF4-dependent manner. And mitochondrial tRNA synthetase inhibitors also extend the lifespan of C. elegans in an ATF4-dependent manner. As mentioned previously, this pathway is conserved in mammals, and there have also been other lifespan interventions that have been associated with GCN4. This paper provides us with new interventions that could be used to further life and health span research. Thank you so much for your time.
Thanks, Shiva. And it's nice to see that data again. Most of that was done by Mark McCormick, who's a former postdoctoral fellow of mine who has his own lab now. We'll have him on the show someday. And it's also nice to see some yeast biology. Charles Brenner has done a lot of work in yeast over the years, so I'm sure it's familiar to him as well. Dr. Brenner is one of the world's leading researchers in the biology of nicotinamide adenine dinucleotide, or NAD, the central catalyst of metabolism. And today he's going to provide a brief but comprehensive review of the NAD system in health and disease. The title of his talk is NAD coenzymes are disturbed in diseases and conditions of metabolic stress, prospects for enhanced resiliency with NAD boosting. It's good to see you, Charlie. Thanks for being on the show.
It's my pleasure. I think I'm sharing a screen now.
Yeah, it looks good.
Fantastic. Terrific. So thank you, Brian, and thank you, Andrea, for inviting me to participate in this symposium. Good early morning to folks of us in the Americas, and good midday and afternoon to folks in Europe, and good evening in the Far East. So I have a few disclosures that are on this slide, let's get off to talk about how things work. So most of us are familiar with electrical devices, whether it's a toaster oven, toaster that plugs into your wall outlet, or an electric car. We all know that electric things require electric power, right? So there's high energy electrons that are running from either the power supply through the wall outlet or through a giant battery in your vehicle. There's copper wires that go from that power supply to all of the component parts of electrical devices that require power. And it's that flow of electrons through copper wires that allows these devices to function. What you might not think about every day is that there's flow of electrons in all of biology. And in fact, the energy for most of the life on the planet actually begins with photons. And I'm not seeing my own pointer here. Whoops. Hang on a second. Do you folks see my pointer?
Actually, I'm not either. It was working earlier.
It was working before, but it's okay. You know, the sun location in this picture is obvious. The energy for most of the living things on planet Earth originates with the sun. The high energy photons from the sun hit the Earth's surface and grow green plants, right? Animals come along and they eat the plants, and they're ingesting protein, fat, and carbohydrates, macronutrients. And then we come along and we either eat the plants directly or we eat the animals that eat the plants, and we run what's called metabolism. And so I won't go through this entire chart, but suffice it to say that metabolism consists of all of the processes that convert everything that we eat into everything that we are. And additionally, a very important point that is emphasized by this diagram on the lower right is that nearly all of the work in biology is done with the energy delta between the high energy of electrons in NADH and as the electron donor, and the level of electrons that are combining with oxygen as an electron acceptor. And so if you look at this water mill picture on the right, the water is coming into the mill at a high potential energy and is being transferred to lower and lower acceptors in a process that turns that mill around. And you can see that turning that water mill can do work. So you can grind, grain into flour with that power. And that is literally the way biology works, is that there's high energy electrons on electron carriers that are transferred to carriers at a lower energy state. And so the key carriers of high energy electrons are called NAD coenzymes, nicotinamide adenine dinucleotide coenzymes. So we have NAD plus on the left, we have NADH on the right. You can see a typical redox reaction in the middle. There's an alcohol being converted to a ketone. This could be malate being converted to oxaloacetate. And what happens is that there is a hydride, which is a proton with 2 electrons, that is captured from a small molecule. Think of it as a food breakdown, protein, fat, and carbohydrate. There's 2 high energy electrons that are captured from the alcohol and put onto the NAD plus, forming NADH. So when we oxidize these fuels that were grown by the solar energy, burning protein, fat, and carbohydrate, we're harvesting high energy electrons onto NADH. And in a reverse process, NADH donates those electrons right into the electron transfer chain at the inner mitochondrial membrane in order to make ATP. In addition, NADH donates high energy electrons in gluconeogenesis and in ketogenesis to maintain blood glucose and to generate ketone bodies in reactions that very much resemble going from right to left of let's say a ketone to an alcohol. Now, if you watch the 2 oxygen on the adenine portion of the NAD, you can see that it becomes phosphorylated. And so there's 2 other key coenzymes in the NAD system. There's NAD plus, NADH, and then NADP plus on the left here, and NADPH on the right. And NADP is the electron acceptor for what's called the pentose phosphate pathway, which is required to make RNA and DNA precursors. So you know that, in the fields of grass or the fields of corn out in the American Midwest, when that solar energy is formed, there's a massive amount of hexoses that are made, right? And we have 5 millimolar blood glucose. So we live in kind of a hexose economy. But what about the information economy of DNA and RNA? That's all done with ribose and deoxyribose. So we make our own DNA, we make our own RNA, and the pentose phosphate pathway is a pathway that every cell knows how to do. And the key electron acceptor for the pentose phosphate pathway is this molecule on the left, NADP. Now, NADPH is a really important NAD coenzyme that uses the electrons that were captured from the pentose phosphate pathway to do free radical detoxification. Clearly something important in life and healthy aging, as well as in lipid and steroid synthesis. So all the testosterone that you have and all the estrogen that you make comes, and all of the lipids are manufactured by virtue of high energy electrons that were captured onto NADP that were donated by NADPH. Now, we got interested in NAD biosynthesis early in this century. We received a lot of received wisdom, most of which was right, a little bit of which was wrong or incomplete, but it was known for quite a while that there's a so-called de novo pathway. So in eukaryotes, you can start with the amino acid tryptophan, which you see on the bottom, and with 6 enzymatic steps that require oxygen, by the way, you can form a mononucleotide called NAMN. Remember, and you can see on the top, NAD is a dinucleotide. There's a nicotinamide mononucleotide that's fused to an adenine mononucleotide. So tryptophan can, through what's called a de novo, a from scratch pathway, generate NAMN. NAMN goes to a dinucleotide called nicotinic acid adenine dinucleotide. And then finally, there's an enzyme that forms NAD. Later, it was discovered that there's a pathway to make NAD from nicotinamide. So NAD can be broken down, and we'll talk more about that soon. The nicotinamide base from NAD can be lopped off. And so there needs to be a pathway that can salvage, that can collect that valuable unit of NAD and form a mononucleotide, in this case, NMN. So nicotinamide to NMN is encoded by an enzyme called NAMPT, and then NMN to NAD is encoded by yet another enzyme. Now, nicotinamide can break down further all the way to nicotinic acid, and the pathway from nicotinic acid to NAD was worked out in 1958 by Jack Preece and Phil Handler. So the so-called Preece-Handler pathway, where nicotinic acid can also be converted to NAMN, just as tryptophan can go to NAMN in 6 steps. Nicotinic acid can go to NAMN in 1 step, and then the other enzymes are the same. Now, I was working, as Brian said, in the yeast system in the early 2000s, in which there isn't this NAM pathway. And so we knew that yeast cells can make NAD from nicotinic acid. They can make NAD from tryptophan. They can also make NAD from nicotinamide, but only by degrading the nicotinamide to nicotinic acid. And I got interested in this enzyme on the upper right that converts the NAAD into NAD, because it was actually an unsolved problem all the way from 1958. of how the ammonia group is put onto NAD to form NAD. So it was known that it's a glutamine-dependent enzyme, but the actual polypeptide basis for that glutamine dependence was not known. So my group cloned the eukaryotic NAD synthetases, and we showed that there is a domain in the protein related to something called nitrulase that is responsible for that activity. So that was a nice study, and we showed that when we knocked out this glutamine-dependent NAD synthetase, the yeast couldn't grow, because you can see that yeast requires that enzyme to make NAD, the central catalyst of metabolism, from either tryptophan or nicotinic acid, and for that matter, from degradation of nicotinamide as well. And we did a lot of cool things with that enzyme. We showed that the glutaminase domain and the NAD synthetase domain both needed to be in the same polypeptide and so forth and so on. But then we decided we're questioning the received wisdom, and we wondered whether there was any additional ways that yeast cells could make NAD. So this was the experiment, and we discovered the identity of the nicotinamide riboside kinase pathway, the existence of the possibility that nicotinamide riboside, NR, could be used as a vitamin precursor of NAD in eukaryotes came from this study. So we knocked out the glutamine-dependent NAD synthetase. We found that nicotinamide riboside could bypass all known routes to NAD through a nicotinamide riboside kinase that we found not only in yeast, but also in mice and in humans. So that was a nice body of work. And so now, at this point in 2004, we have all the biosynthetic pathways for NAD in eukaryotes. And let's just turn to kind of finish the introductory information to what are called NAD-consuming enzymes. Because after all, it's this turnover of NAD to the nicotinamide product that I explained to you is required for the use case for some of these NAD biosynthetic pathways. So what kind of enzymes and biological processes are involved in degradation of NAD? I just told you that NAD coenzymes are the central catalysts of metabolism. Can't live without them, can't make ATP without them. Yet there are enzymes, and I'm going to call them pathway A, B, C, and D, that will break down the NAD. And I hope that you can see that NAD is broken into a nicotinamide piece, nicotinamide moiety, and a piece that contains an ADP ribose unit. And in this first case, an ADP ribose unit is basically stuck onto a polypeptide, usually a serine or aspartate residue. So this is called mono-ADP ribosylation. And just to show you some of the recent work that our group has done on mono-ADP ribosylation, we found early in the COVID pandemic that in cells that are infected by SARS-CoV-2, there are 5 nearly uncharacterized enzymes, members of the so-called PARP superfamily, but enzymes that do a mono-ADP ribosylation reaction, namely PARP7, PARP14, PARP13, PARP9, PARP10. You can see them in the upper right of this volcano plot. 5 members of the PARP superfamily are transcriptionally induced by a very great degree upon coronavirus infection. And we showed that, in fact, coronavirus infection attacks the NAD system by virtue of transcriptional upregulation of these mono-ADP ribosylating PARPs. It turns out this is a common theme in viral infection and inflammation. This work has been translated. There are multiple clinical trials ongoing involving nicotinamide riboside. In fact, there were successful phase 2 and 3 trials showing that an NR containing cocktail accelerates time to recovery from COVID. And there's multiple registered clinical trials that are in this space. So NAD can come under attack from mono ADP ribosylating enzymes, largely due to transcriptional upregulation. And this is downstream of the innate immune system identifying double-stranded RNA in a cell. Now, famously, there's pathway B, which is called poly-ADP ribosylation, where after one ADP ribose unit is installed onto a amino acid side chain, and enzymes called PARP1 and PARP2 can install multiple units of ADP ribose. And they can actually form poly-ADP ribose that is not linked to a polypeptide as well. And this is also highly induced and highly specific for specific biological signals, like DNA, single-strand DNA damage and reactive oxygen species will activate PARP1, and you can see it will form these poly-ADP ribose modifications on amino acid side chains, as well as PAR formation in the nucleus and cytosol as well, and activate all sorts of biological processes, ATP depletion, calcium mobilization, and the like. And this is actually essential for aspects of single-strand DNA repair. And a third pathway is cyclic ADP ribose formation, where the NAD gets cyclized with elimination of the nicotinamide unit. And one of the enzymes, there are two principal enzymes that do this. One is called CD38, which we've done a fair bit of work on, and another is called SARM1. And here's an illustration on SARM1 from the Michael Coleman lab in the UK. And essentially, SARM1 is activated by NMN, by accumulation of NAD biosynthetic intermediate NMN, so that when axons are injured or the NMN-AT2 gene is mutated, or the NMN-AT2 protein is eliminated by axon injury, and with particular drugs and with particular other mutations, NMN accumulates and activates SARM1 that leads to formation of cyclic ADP ribose and degeneration of neurons through a process called axonopathy or Wallerian degeneration. Finally, there's the reaction that most of you have probably heard about, which is NAD-dependent protein lysine deacetylation. So some acetyltransferase has put an acyl group, typically acetyl, but sometimes propionyl, butyryl, or some other longer chain acyl modification on a lysine. And then in an NAD-dependent fashion, an enzyme related to SIR2 comes along and deacetylates, again, forming a nicotinamide unit and then an acetylated ADP ribose. Now, a lot of ink has been used to characterize these sirtuin reactions. And for example, SIRT3, which is a mitochondrial sirtuin, has been claimed to activate dozens of different enzymes. But it turns out it's kind of a strange hypothesis, because SIRT3, all SIRT3 does is remove acetyl modifications that some other process puts on. So SIRT3 has no role in where the acetyl group goes on to a polypeptide. And we showed in 2013 that it's actually just mass action, and the intrinsic pKa of particular lysine residues that controls where the acetyl group goes on, which lysines get acetylated, and then SIRT3 comes along and just cleans up these acetyl modifications. So SIRT3 is more of a garbage person, and garbage people are very important in society, cleaning things up. But SIRT3 is not a highly regulated enzyme, and neither do we think are many of the sirtuin enzymes, despite claims to the contrary. So to summarize here, morylation is highly induced by infection, is clearly druggable. You can attack these morylating enzymes themselves. You can use nicotinamide riboside to counteract the NAD depleting effects of them. PARylation is highly induced by DNA damage. There are approved drugs that target PARP. SARM1 is highly induced by NMN, also druggable, at least in principle, on the basis of genetic studies. And then we have the sirtuin thesis, which in my view has been one of the biggest strains on biomedical research in history. And I've done a fair bit of writing on this. And just to clarify, the original result in which the yeast SIR2 gene was claimed to be a longevity gene, it came from an assay that we just heard about earlier in this hour, what so-called replicative lifespan. So this is what a budding yeast cell looks like, this thing with a mother cell producing a daughter. And if you look in a culture like in Flask B, in a culture of yeast cells, half of the cells are new daughters. They've never been a mother, right? And a quarter of the cells have been a mother one time, and an eighth of the cells have been a mother two times. And so very, very few cells have been a mother many, many times at all. But in the yeast replicative lifespan model, one installs a graduate student or a postdoc in front of a dissecting microscope at the beginning of a 2-week period and arrays new mother cells. We've done it in our lab. Brian Kennedy learned it when he was a graduate student. And you literally just take the new daughter cells and throw them in the trash, and you keep track of how many times the mother can produce daughters. And the result was that if you have more copies of SIR2, then you can extend lifespan in most experiments from 21 generations to around 26. But if you do the math, that means only 1 out of 5 million cells has an advantage of having extra copies of SIR2. And when Walter Longo did a yeast chronological aging experiment, he showed that if you delete SIR2, you get longer chronological lifespan. This is actually a selected trait. It's not 1 out of 5 million cells having an advantage. It's all cells in the culture being better off if they don't have SIR2. They actually live for more time. And so in this comprehensive review article, we've shown that the SIR2 extends lifespan in 1 out of 5 million cells. That seems to be factual, although it's genetic background specific. SIR2 deletion extends lifespan, where extra copies of SIR2 shorten lifespan in the chronological model. You probably are aware that there are papers that were published that were cited thousands of times, claiming that in worms and flies, extra copies of SIR2 extend lifespan, but that was debunked in a paper from David Gems and Linda Partridge in 2011. And in fact, deletion of 1 copy of the fly SIRT1 gene extends lifespan in flies. Not very many people even know about that paper. And then 4 groups did a whole body SIRT1 overexpression with no life extension. And 1 group did a brain-specific overexpression of SIRT1, which did not consistently extend lifespan. And SIRT6, which is not SIRT1, but people have been all over this entire gene family, in some mouse backgrounds in males, and maybe in some mouse backgrounds in both males and females, lives longer. That's true. But by promoting higher gluconeogenesis, which is good for an older mouse, but it's really bad for an older person. And so there's inaccurate information in hundreds of review articles that have been highly cited. There are thousands of research projects that I believe have been mispremised and suffer the consequences of bias, hype, and forming a hypothesis after the results are known. The resveratrol story of being an activator is wrong, and as constructed is non-falsifiable. I can tell you more about that if you're interested. And so the implications for popular expectations of having a dominantly acting longevity gene, which SIR2 is not in mammalian cells, are enormous. And that's a problem. And there's a huge bias in my field that sirtuins mediate the effects of NAD modulation. So now I'm going to show you some data from our lab and others showing what happens with NAD modulation. We developed a technology called quantitative targeted NAD metabolomics. So we have labeled internal standards of the NAD metabolome, and then we can find in a lot of disease models that the NAD system is disturbed. That was in the title of my talk. And this is in a model of dilated cardiomyopathy, a model of heart failure in mice, in which the serum response factor is deleted from cardiomyocytes. And what you can find in this cardiomyocyte model is the NMRK2 gene, the nicotinamide riboside kinase 2 gene in the mouse, is the most highly induced gene in the failing heart. And this NAMPT gene that is responsible for nicotinamide salvage, its expression is going down. So that failing cardiomyocyte seems to want an upregulation of the NR pathway while it's degrading expression of the nicotinamide salvage pathway. And you actually see this, as you can see, in human heart failure, not only in mouse heart failure, but in human heart failure, that the NAMPT level expression is going down and NRK2 expression is going up. So oral nicotinamide cannot restore a cardiac NAD in this model. Nicotinamide riboside can't, and nicotinamide riboside can preserve the left ventricular ejection fraction and the fraction shortening and 6 other things that we measured in this model. So nicotinamide riboside is preserving cardiac function and vitality in this mouse model. NAD is under attack. The NRK pathway is upregulated and NR is highly protected. So in another system in which the NAD system is disturbed, we looked at the mother of all metabolic stresses, which is postpartum. And so we found in the system that in a postpartum, that means a new mother mouse or rat who is lactating, there's lower hepatic NAD. That new mother mouse is circulating her NAD metabolome, and she has higher blood NAD metabolites and lower hepatic NAD metabolites. So she's not only mobilizing protein, fat, and carbohydrate from her liver and adipose stores to her mammary, she's actually mobilizing NAD metabolites from her liver to her mammary, where her mammary NAD system is induced by 30-fold, as you can see in the lower left. And this has remarkable consequences for the offspring. The mouse in the bottom panel is the offspring of an NR supplemented mother. The mouse on the top is the offspring of a normal mom. They can both get across this balance beam, but you can see that if you supplement the mom, their offspring are much better off. And we have a beautiful paper on this that we published a few years ago that showed that that juvenile offspring of NR supplemented mothers are more resilient, more adventurous, and more neurodevelopmentally advanced. They actually preserve brain functions into their adulthood, and they're advantaged in hippocampal neurogenesis. And I encourage you all to read that paper. So to summarize, the conditions that we found challenge NAD, we found in human liver samples that alcoholics have a lower NAD system. We found in mouse liver samples that overfeeding mice depresses their hepatic NAD pH. Anthony Sauvé, the late Anthony Sauvé's lab, showed that noise sufficient to produce hearing loss attacks cochlear NAD. We and others found that sun and oxygen damage disturb the NAD system. Changing time zones and irregular hours, circadian disruptions disturb the NAD system. Infection and inflammation, I showed you a little bit of those COVID data. Mitochondrial disease with Anu Soumalian, we showed that people with mitochondrial disease circulate lower NAD levels. Also, neurodegeneration, that's the SARM story in part. Heart failure, I showed you some of those data. Pregnancy and postpartum, I showed you some of those data. And then it's widely reported that NAD system comes under attack in aging. So as you probably know, our discovery of the NR kinase pathway and the activity of NR as a vitamin has been commercialized. We've done safety and testing of NR in many studies. This shows dose-dependent increases in human blood NAD with the safety-tested commercialized nicotinamide riboside, which is called Niagen. And there are hundreds of animal studies showing beneficial effects of nicotinamide riboside, and there are now 79 registered clinical trials that have been done with NR or NR in combination, including 7 human replications showing anti-inflammatory activities of nicotinamide riboside in human beings. The best indications for nicotinamide riboside are when the NAD system comes under attack. Ideally, when the NMRK1 and or NMRK2 genes are being induced. If you look at that system in a preclinical model, or ideally in a clinical model, you see the NAD system come under attack and the NR kinase expression system is high. There's a reasonable likelihood of success that oral nicotinamide riboside is going to be effective. We think that heart failure is one of those indications. Inflammation has been replicated, and fatty liver or metabolic dysfunction-associated fatty liver disease is an important one. And we plan on testing skin healing from shave excisions. Why? Because in older in life, you heal cuts and scrapes slower, and you can watch the entire time course of healing in an older person versus a younger person. You're not looking at lifespan extension, but a boosting of resiliency with nicotinamide riboside. And so to summarize, 4 NAD coenzymes are the central catalysts of metabolism. Their essential function is carrying high energy electrons. 3 of the 4 NAD systems, namely mono ADP ribosylation, poly ADP ribosylation, cyclic ADP ribosylation systems are intensely regulated. The sirtuin system seems to be a rather kind of tonic system that is always operating. It's been intensely investigated. I think it's been intensely hyped with relatively little to show for the amount of attention that it's gotten. Quantitative targeted NAD metabolomics has allowed us to identify many conditions in which the NAD system comes under attack. And the NRK system, largely through NRK1 being expressed in every cell at a constant level, is constitutive, while the NRK2 gene is inducible and is a very good signal that a tissue is looking for NR in order to maintain bioenergetics and cellular function and resiliency. And so with that, I'd be more than happy to take people's questions.
Thanks a lot, Charles. We're a little over, so I'm going to try to ask some questions quickly. First of all, I wished you were teaching biochemistry to me as an undergraduate. It would have accelerated my learning. That was a good lecture in the basic pathways there, metabolism. I got to push back a little bit on the yeast replicative assay, not so much on the sirtuins, but a couple reasons. One is that the argument, as I took it, was that you're talking about one in millions of cells, so how could it matter? But most of the argument about aging itself is that it's what happens after natural selection declines. And so making an organism, a human, live longer is not clearly improving its fitness either. So I'm not so sure I understand that argument. And beyond that, let me do one more thought, and then I'll let you have your time. Then we showed pretty clearly that by looking at the whole genome in yeast and comparing that knockout to the whole genome in worms, there's conserved longevity pathways going on between these two very disparate systems, which has never been shown in biologic aging. So,
right. So mTOR, for example, right? mTOR, pathway mutations extend lifespan in the replicative model and the chronological yeast model, and then are found to have activity in animals. But the sirtuin pathway, which fails, right, in animals, only has an advantage for this one rare mother cell in the replicative model. It shortens lifespan in the chronological aging model, and it has no reproducible effect in animals. So it's just simply a fact.
Not just score, though. It's quantitative evidence for replicative aging being conserved with worms, with tens and almost 100 genes. So it's beyond.
Sirtuin is not one of them.
I'm not debating the sirtuin argument, but I think it is a valuable model. Nonetheless, SIRT6, does seem to extend lifespan. Does NAD even activate SIRT6? It uses NAD, I understand, but can you measure enhanced SIRT6 activity with any NAD precursor?
NAD precursors maintain the NAD system.
Yeah.
And so there are, just in the PARP family, there's 15 or 17 members of the PARP family that depend on NAD. There's CD38 and SARM1 that use NAD. There's 7 sirtuins that use NAD. And then there's dozens of redox enzymes that use NAD, NADH, NADP, and NADPH. So when you elevate NAD levels, you can measure increased activities of many different enzymes. But the evidence that specific sirtuins mediate the beneficial effects of nicotinamide riboside are deeply lacking. For example, in the initial paper from Kanto and Auerks on nicotinamide riboside benefiting overfed mice, what they did is they blotted for the acetylation state of P53 and SOD2 or some mitochondrial enzyme. And so you can see, and they said that was consistent with SIRT1 being, quote, activated and SIRT3 being activated. But that's not a genetic experiment saying that the beneficial effects of nicotinamide riboside were mediated by either of those enzymes. Yeah. Just the fact that you see higher levels of enzyme is not a genetic test. So, for example, in our systems, for example, in heart failure and in promoting weight loss of new moms and the physical fitness of their offspring, we believe the beneficial effects of nicotinamide riboside are highly pleiotropic.
Yeah, I
And
agree. I'm not arguing that it was through sirtuins. My question was, I was really arguing more your point, I think, than that, which was that I haven't even seen how SIRT6, how much responsiveness there is in SIRT6 to NAD levels in the cell.
well, but see, those experiments were done with a model bias that that thinking that sirtuins were conserved longevity genes. We know that the papers on extra copies of SIRT, extending lifespan in worm and fly were not reproducible when the strains were cleaned up, by in 2011, but people went off with the expectation that sirtuins were going to be dominantly acting longevity genes, and so they tended to report that sirtuins were dominantly acting longevity genes.
Let me turn
Even
to
if they
let me
didn't
just put it a
find it
different way.
like in SIRT6, if they didn't find it in one mouse strain, they would literally cross it into another strain until they found it. And when you see authentic, there are monogenic longevity syndromes in animals, right? Largely in pituitary pathways. So in the insulin-like growth factor system in worm, and in the entire growth hormone receptor pathway in mice, it's inactivation of these growth promoting and maturation promoting pathways that gives you very long-lived worms and very long-lived mice. But that's a different story than claiming that something is a gain of function mutation that extends lifespan. And so I don't believe that there are any gain of function mutations in animals that extend lifespan.
There are definitely in yeast and worms gain of function mutations. I don't know if there's not much in mammals that have been shown so far, I think. But anyway, I don't
believe that there are any dominantly acting longevity genes in vertebrates. If there were, the implications for CRISPR would be profound, right? Because if we found a dominantly acting longevity gene, then we would all want more copies of it, which is kind of
not if it does. It may have
one thing.
It may have a fitness defect, though, due to antagonistic pleiotropy that would make us not want it. Anyway,
I'm not aware of any dominantly acting longevity genes in vertebrates.
Let's talk NAD, because I think that's what people want to hear about. So if it's not going through sirtuins, and I agree with you that I don't think there's much evidence that it is, what do you think it's doing? Is it just sort of metabolic health of the cell, and many other pathways are being preserved when you have NAD level?
It's tissue repair. It's responding to these conditions of metabolic stress. So essentially, the message of our laboratory from the last 20 plus years of working on NAD is that NAD coenzymes are the crown jewels of metabolism, the central catalysts that carry high energy electrons that make all things possible in living things, including lipid synthesis, DNA repair, and resiliency to reactive oxygen species. We find, surprisingly, that the crown jewels of metabolism are not locked inside of a safe, put inside of a vault, patrolled by armed guards and a moat, but in fact, the crown jewels of metabolism are exposed to the elements of metabolic stress. So ultraviolet light that damages DNA will end up attacking the NAD system. Reactive oxygen species, we live in an oxygen world, to generate ATP, challenges NADPH. And so many different conditions of that are common in life, as well as disease processes like axon damage that accumulates NMN and activates SARM1. These things disturb the NAD system. And that when you see upregulation of NAD biosynthetic genes, there's a very likely use case for NAD precursors in maintaining resiliency. But the idea that it's mediated by one particular NAD-dependent enzyme, I think, is naive and mispremised.
We could talk all day. I mean, I agree with that statement. I think that let me bring in Jonas though, because there are a number of questions from the audience.
Great.
Yeah.
Okay, thank you, Brian, and thank you again, Prof Brenner, for the presentations. We have a few questions, particularly on supplementation. So I think I'll start off with the first one.
I didn't ask Jonas because I knew the audience was going to.
Yeah. Maybe I'll continue on that. The question is, should we be taking NMN, NAD, and vitamin B3 orally, or should these be taken by IV drip to recover the lower levels due to aging?
Okay, NMN and NAD. So NMN has a phosphate, NAD has 2 phosphates, NADPH has 3 phosphates. Compounds with phosphates don't get into cells. Accumulated NMN in cells activates SARM1. So it doesn't actually even make any sense that people would think that NMN gets into cells. So cells have the ability to take up either of the NAD precursor vitamins, nicotinic acid, nicotinamide, or NR. And nicotinamide and NR can largely be used by any cell or tissue in the body. As I showed you, there's many conditions of metabolic stress in which the NR pathway becomes more important and becomes upregulated. These are all orally available vitamins. Oral supplementation of NR is very effective at elevating blood NAD, as I showed. Its ability to elevate tissue NAD is being demonstrated in trials around the world. There is now a published patent application from the group in Norway showing that oral nicotinamide riboside elevates brain NAD in the context of Parkinson's disease patients, and also showed beneficial effects on cerebral blood flow and I believe cognition in that population. So oral nicotinamide riboside is effective. People have started doing things like intravenous NAD. I understand that it's painful. I believe what it's causing is an innate immune response because NAD is normally an intracellular metabolite. When cells see things like double-stranded RNA, intact mitochondria, cell-free DNA, ATP, and NAD, it kicks off an innate immune response. As the NAD breaks down extracellularly in the plasma into NR, it gets into cells. So I don't believe that there's really a use case for intravenous NAD. It doesn't get into cells as NAD, and it doesn't get into cells as NMN.
Thank you, Prof. Brenner. And the next question is on resveratrol. It's always been voodoo science that resveratrol is the explanation for the health benefits of red wine. It's insignificant or absent from most red wines. So how do we get the wider world to recognize the snake oil of longevity science?
I strongly recommend my review article, Sirtuins Are Not Conserved Longevity Genes. I have a section on resveratrol in there. Resveratrol does not hit the SIRT1 target that was shown to be a biochemical artifact within a year or two after the original reports from Howitz et al. SIRT1 is not a target that would want to activate anyway on the basis of much of the data that I've showed. Unfortunately, there is a great deal of hype in the space.
And the next question is regarding the dosage. What amounts of NR should we be taking daily in milligram?
I believe that there is a use case for oral nicotinamide riboside. As you've seen from my seminar, tissues like the liver can make NAD from tryptophan, but in many of these conditions of metabolic stress, many of which are inevitable, like oxygen and sunlight, our NAD system comes under attack. Our data show that even at 100 milligrams, there's some noticeable rise in blood NAD. Tissue NAD is harder to, quantify because people aren't going around and donating, me pieces of their brains. Clinical trials are largely being done at a gram of nicotinamide riboside per day. Oral supplementation, the typical consumer dose is 300 milligrams, but there are, professional athletes and certainly people in clinical trials that take higher doses like a gram per day.
I think the next question is regarding wound healing. You've mentioned that NR is also helpful in wound healing. The question is Sanjay is wondering, is there any study that has been reported that is also helpful in organ transplantations or organ tissue regeneration like liver?
I think it is an active research area. There's not a randomized clinical trial, to my knowledge, in that space. In our case, there are mouse data showing that if you depress the NAD system, you get poor wound healing in mice, and that if you supplement those mice with NR, their wound healing is better. And so we've set up proposed a randomized clinical trial with people of varying ages from 18 to as old as we get them at City of Hope in Los Angeles to characterize skin NAD system quantitatively in human beings, as well as to test the efficacy of NR against placebo in promoting wound healing.
The next question is there a diurnal variation in NAD and NADH ratio and concentrations as part of a daily nutrient regimen?
In mouse, in mouse, we have a paper with the late group at UC Irvine, Sassoni Corsi, that showed that there is circadian variation in the NAD system, and it probably is diurnal.
Okay, the last question is regarding stem cells. So do you think stem cell will help women who's been diagnosed with premature ovarian failure? How will the stem cell regenerate ovary tissue? If it does help, how often do we need to repeat the stem cell therapy? And is there any specific point to inject the stem cell?
That's not even a question for me.
No, but there is data on NR and fertility in mice, I think. What do you think of that data?
We're the ones that got into maternal health, and we think that when the NAD system is disturbed, that there's a use case. In human, follicular maturation, there is a time course that is, quite severe in terms of the ability of primary follicles to, develop into, functional oocytes. And I don't think that nicotinamide riboside as a monotherapy or monoprevention agent is going to entirely solve that problem. In terms of stem cells, into that, I mean, that's a highly speculative area.
So thanks a lot, Charles, for coming on the show, especially so early. We'll have to have you back sometime. Thanks, Jonas. This is your first show of many, I hope, and you did a great job. So I want to remind people to use the chat function, and the panelists and all attendees option to leave comments and feedback on the show. We have a conference on supplements in the end of February, February 29th and March 1st of next year. One of the first global conferences solely focused on supplements is geroprotective interventions, and there are going to be a lot of different kinds of experts there to discuss the topic. This is my last show of the year, so I want to wish everyone a happy rest of the year and New Year. I'll see you next year, but we do have more shows coming up. Andrea Meyer will be doing the shows in December. The first speaker will be Sachin Panda, who'll be talking about circadian rhythms and aging. And so please come back for that show. Thanks for joining us. And I'll leave you with the final video about a grandmother with a black belt in Taekwondo.
Hello, I'm Lin So. I'm 77 this year, and I have a Taekwondo black belt. Actually, joining Taekwondo is by chance. A chance upon when I came here for a shopping. I saw all those, they're having a warm-up. They call it the facial warm-up, and it fits me very well because I have leg problems, knee problems in fact. My problems was can't squat, pain, every morning pain from the sciatica. The leg all went down to my foot, my feet like both, and I was so miserable. So after joining Taekwondo, oh, that my happy days. It just disappeared, all the pain. And my orthopedic has to reduce all the medication and say, oh, no more pain medication for you. You are okay, you are fine. But I think when you age, you need a lot of movement to your muscles. The advice is, please don't wait. No, go for it. Don't say no point. The more you say it, the more you lie on the bed and look at the ceiling. Do you think you want this style of life? No, right? Mobile, very important. Our 2 legs, God gave us 2 legs, must move.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.